All right, so I've started recording. So on Friday, I think I realized that I didn't upload. I recorded the video. I don't think I uploaded it until this morning, so my apologies on that. So if you haven't watched the lecture from Friday, give that a go. That one basically focuses on the structure of DNA, and what we'll be spending on the lecture for today is going into RNA, and then also how proteins are transcribed from RNA. So that's going to be our main goal for today. So some of these slides we've already seen if you were here on Friday, right? So we're just going to briefly talk about three different types of ribonucleic acid. So three different types of RNA molecules. So they'll be made out of the same building blocks like we talked about before, with a base and a sugar and a phosphate backbone, right? But these are going to have distinct roles inside the cell. And so the three that we're going to talk about are rRNA or ribosomal RNA. And so these are going to be responsible for building a large structure that actually makes proteins. So you might have heard of ribosomes before. We'll also talk about messenger RNA, and that one actually makes probably the most sense because it's in its name, right? This is going to be the RNA that takes information from DNA and then gets it all the way to the ribosome to make protein. And then the last type of RNA we'll talk about is tRNA, which is transfer RNA. So this is the RNA that's bonded to amino acids, and it will bring those amino acids to the ribosome where it actually is synthesized into a protein. So we'll go step by step through each of those processes. So we'll start with the easiest one. This is structurally, it looks different, so it kind of sticks out, is tRNA. So you don't really need to worry about the structure so much, though you can see it's distinctly different than the double helix of DNA that we talked about before. But there's two main parts of tRNA that are really defining its function, right? The first is going to be this area up here. There's nothing drawn here, but this is where the amino acid would be bonded to. So it'd actually make a covalent bond with that hydroxyl group, and that tRNA is basically going to float around connected to that amino acid. And we'll see in a couple of slides how that tRNA then donates that amino acid to make that polypeptide chain that eventually becomes the protein. The second most important structure on a tRNA molecule is going to actually be down on the other end of the molecule, where you see this loop down here. And this loop is called an anticodon. So the word codon, what it's just talking about is in DNA, right? We know it's made out of A, T, G's, and C's. And the way that your body stores information and then your DNA actually tells, a ribosome how to make the protein is that there'll always be a sequence of 3 amino acids that are in the DNA. And those 3 amino acids then will match up, but it will be the opposite. So if it was like ATG, the opposite of ATG, which we talked about on Friday, would be TAC. Right, so it's the anti of the 3 set codon that would be in DNA. So long story short, it's basically kind of like it's recognizing where to plug in that amino acid. So it's just recognizing the DNA sequence. And again, we're going to spend a little bit more time on that. So if it doesn't feel super comfortable, we're definitely going to get comfortable with it. So those are our 2 main take-home structures of tRNA. Right, so the other RNAs, we didn't look at the structure, so that would have been the ribosomal RNA and then the mRNA, because it's just RNA. It looks like DNA, but it's single-stranded, right? But they have distinct functions. So how does your body take the information that's stored in DNA, right, and then get it all the way into a protein, right? There's going to be 2 distinct steps. The first is going to be transcription. Oops. Let's do the whole thing here, right? Transcription, and that's going to be from DNA to RNA, specifically messenger RNA. And that messenger RNA is then going to be translated into that protein structure, or that polypeptide, right? And this will be done through the use, let me make this a little bit more structured, right? And this is going to be using tRNA, which then when it goes through the ribosome, which is made out of rRNA, right? It will then be transcribed into a protein. So it can get a little bit complicated, but the process doesn't change. It's always going to follow this path that DNA will be turned into mRNA through the process of transcription. The mRNA will be read and translated by the ribosome, which is made out of our RNA, using little bits of molecules called tRNA that when put together into the ribosome, the protein will actually pop off. So those amino acids on the tRNA actually will come off the RNA and just start making a long chain of protein. So I don't think the book gets into this. An old way of referring to this whole process was central dogma. And so that's another way you might hear people refer to this particular set. This is, this set of steps. So all organisms that we know of follow these steps. Therefore, that's why we call it central dogma. It explains how we get from DNA or information stored in DNA to an actual functional molecule as a protein. And so this is more just like a picture version of a little sketch that I did before, right? So the DNA, there's our double-stranded helix. It's going to be transcribed into mRNA. The figure here, you can start to see, it would not actually look like this in real life, but it's starting to group together, looks like 3 little dots, right? Those are the codons that the anticodon on tRNA will recognize. And then when it goes to the ribosome, well, there's my long chain of amino acids. So how does this actually get done, right? So it's not magic, and it's actually done by a bunch of other proteins. And so this way, a lot of people get into that chicken or the egg type question. Well, if DNA can't, without proteins, DNA can't make more proteins, right? So which one came first? Did proteins come first? Did DNA come first? Actually, the theory is that RNA alone came first, and then after that, DNA and proteins evolved from there. That's beyond the course. But it can get a little bit tricky saying, okay, well, how does, if I don't have proteins to start with, I can't do the whole, I can't do this process. So it gets a little complicated. But we're going to keep it really simple. There's actually a tremendous amount of steps that happen. But in general, we'll just talk about one type of enzyme called an RNA polymerase. And so you might have heard this term before, especially if you've heard the types of tests that some of the types of tests they use to test for COVID-19 uses something called PCR, which is a polymerase chain reaction. It doesn't use RNA polymerase, it uses another molecule called DNA polymerase. But if we break down what the word means, polymerase, we can see that it has a broad definition. So a polymerase, it just says poly being many, pieces, right? And it's saying it's the substrate of an enzyme. Specifically, it's going to be many pieces of RNA. So what ends up happening is you have this big strand of DNA, an RNA polymerase comes by, which is going to be represented by this orange kind of ellipsis, right? So that's why you see RNA polymerase down here. It's just saying it's a big bulky enzyme. It literally pulls apart the DNA, and the enzyme starts reading the DNA and then starts making its own chain of RNA based in the exact opposite sequence, right? So if it was ATG, it'd be TAC, right? And so we're synthesizing this new strand of single-stranded mRNA. And this is all done in the nucleus, right? So again, getting a little bit deeper into this idea of codons and anticodons, right? This is what we're referring to when we mentioned the genetic code, right? So even though we know it's made out of 4 different base pairs, right? Adenosine, guanine, cytosine, and thymidine, right? With uracil being the T analog in RNA. The information is stored in 3 sets of repeating, or just 3 sets, wouldn't necessarily be repeating, but 3 nucleotides in a row. And so what we see down here, right? We'll make it really simple. The example here is just everything's uracil. So if my RNA sequence was just UUU, when that goes to the ribosome and gets transcribed, the tRNA that calls for phenylalanine would be AAA, 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 right? That would be attached to those. Let's see if I can draw this. right? It would do something like this, where the anticodon, I'm trying my best to draw a tRNA, right? Something like that. And I'm writing PHE because that's the 3-letter code for phenylalanine, right? And so what that your body would recognize is those tRNAs would go in order, would just be 3 phenylalanines in a row, and they'll covalently link and then make a new protein. So it's actually, even though we have those 4-letter codes in DNA, it's the arrangement of all 3 of them in a row that really is going to dictate what amino acid comes next. And so what we have here is something called the codon chart. What it's telling me is, and it's an interesting way to read it, but you get better at it the more you use it. What you're looking for are combinations of a first letter, second letter, and third letter. And that's going to tell you what amino acid would come from that codon, right? So if I was looking for something like histidine, right? I know histidine, I've just done this because I've done it a bunch of times. One of the codons for histidine is CAT if it was in DNA, or it would be CAU if it was RNA, right? T and U being, or U being the analog of T for RNA. So the way I would read that would say my first letter is C, my second letter in this case would be A, and then my final letter would be U, so I'm in this white shaded region, follow it down from A, and there's my U, and it's exactly what I see, CAU. If that combination comes up in an mRNA, your body knows to put in a histidine. And you'll actually see there's a lot of redundancy inside this chart. Like there's more than one codon for histidine. There's more than one for asparagine, right? If you go through, nature has partly, it's kind of randomly evolved to what it needs, is that some amino acids are just used more than others. So there needs to be more codes for those because you could actually use up your supply of tRNA. So having more than one codon for a common amino acid is your way that your body doesn't run out of building blocks. Because if you do, you actually can stop synthesis early and the protein kind of just gets ejected and it's a non-functioning protein, and your body will usually go and then destroy it and then try again. There's other codons in there. You might see the word stop. That's actually termination sequences. And if your body reads those, like UAA or UAG or UGA, your body actually knows to stop making a protein when it reaches that, and then it will eject the protein. Protein is in the length that it's supposed to be. It goes off and does its job. And there's also something called a start codon, which also encodes for methionine. So there's other information that your ribosomes can pick up on, but usually a protein sequence will start with methionine or AUG. It doesn't necessarily mean the methionine will be the first amino acid. A lot of the times it gets cut off, but that's how it starts. So your body has a pre-programmed way to where to start, how to build it, and then where to stop. And it's all that is held inside the information in the DNA, which gets translated to RNA, which gets transcribed finally to protein. Oops, I'm in annotation mode. There we go. Right, so this is more of just like another example instead of just having UUU, right? If we went slowly through this, I could give you a sequence of, in this case, there's 4 separate codons, so the DNA sequence of CCU, AGC, GGA, CUU, right? You could go through using that chart on the last slide and just slowly go, what's my first code? What's my first letter? What's my second? What's my third? And if we went through that, right, we could find out that CCU means proline. AGC is serine, GGA is glycine, CUU is leucine, right? And so when I put those in order, when the protein that pops out, pro, ser, gly, leu. So let's talk a little bit more about what's going on inside the ribosome itself, right? So we have a very similar picture that I used for this section, which is protein synthesis. So we had our tRNA before, right? Remember that we were missing something? And that's our serine amino acid sitting at the top of that tRNA. So when your body needs to actually, it's called charging. So it needs to charge the tRNAs. And what this will do first is it finds free serine floating around in your body. and an enzyme will come along and then actually physically connect it to the tRNA. And now that's called charging or initiation. And then once that is charged, it's ready to go into the whole system, right? So I know this slide's a little bit busy. Maybe I should have made this a little bit bigger. But if you go through slowly, right, it's actually taking us stepwise through everything individually, right? So we're starting in the nucleus, right? I know it's a little bit hard to see, right? But that, what we're looking at here, that purple one, right, the double-stranded nucleic acid strain, that's our DNA, right, the purple one. You can see that it's been unzipped, and then this pink mRNA is being synthesized by RNA polymerase. It will literally snake its way outside of the cell, and it's totally random, right? A lot of these things, for the most part, this is random. There's other places in your body where they can modify things to send them to different organelles, but the RNA just literally snaking its way out. What I have here, and again, sorry, it's hard to see, that says ribosome. Right, I said this is basically the protein making machine. And it's primarily made out of rRNA, though there is some proteins components in it as well. What you see in the figure, we don't get into it. Looks like there's a purple subunit and a blue one. They're called the large and the small ribosome. You need both of them to kind of click together for everything to work, right? So we're feeding our mRNA through the ribosome, right? We have our tRNA, so these little balls up here are amino acids. You charge your tRNA. The tRNA goes into the ribosome. Now everything's all coming together at once, right? We have our mRNA, our rRNA, and our tRNA are all working together. So as you feed the mRNA through the ribosome, right? These tRNAs are making their way in, and then you've actually, out the top of the ribosome, you're literally popping out a protein, one amino acid at a time, until you hit that stop codon, and then the protein itself is ejected from the ribosome, floats off, and it does its job. So I know that we just went through that pretty quick. The reason for that is if we were here on Friday as well, we watched 2 movies on this. This is more just like the me talking through it part. I don't know if YouTube is gonna flag me on that or not. It could be copyright infringement. If it is, what I'll do is I'll remove that part from our video, and then I'll just email you guys the link to those 2 videos. So how are we doing so far? Any questions, anything we need to clarify? Right now, I'm just looking for big picture ideas. All right. So let's talk about when things go wrong in that process, right? So a lot of times people hear the word mutant or something like that, immediately think like X-Men or like, I don't know what goes through people's head when they hear mutations, but nature, the only way evolution works is through mutations, right? So why don't we get an idea of what mutations are and what mutants are? So it sounds cool that, you know, I've made 100 mutants in my life or more. It's not that cool. It sounds cool, way cooler than it is, right? So why don't we talk about what a mutation is? All it is a change in that nucleotide sequence of DNA. So maybe it was AAAA and it changed to AAAT. That would be considered a mutation, right? So why does this result in a change to the body, right? Well, we know that when you're reading off the genetic code, they all come in bundles of 3, right? Those are our codons. So if you switch one nucleotide in a codon, right, you can change the entire sequence. So when I go back a couple of slides to show you what I mean, right? So imagine we did have that, what did I say, CAU, we said was histidine, right? So there's my CAU, right? So let's see what happens if there was a mutation now and say it's CAA. So all I did was change one nucleotide. Well, the first 2 are fine. I have my C, A, but now I'm at A, CAA, that's glutamine, right? So that would be a mutation. That would actually change, it could change the entire structure and function of a protein. And usually mutations are detrimental in the sense that if it is a large enough mutation in the DNA, the cell will kill itself. Other times it sticks around, and that's actually how you can get evolution to happen. It's also how you get genetic diseases and also how you get cancer. So that's why, you know, we're not perfect. Actually, evolution has designed organisms to not be perfect because mutations do lead to genetic advantages. So that's why your machinery is not perfect, because it is designed to evolve. But having that, you know, that opens up kind of Pandora's box that does allow for negative mutations, right? So these can affect structure and functions of the proteins in the cell. So things that can cause mutations are X-rays, because what they can do is they can damage the DNA base pairs themselves. What usually happens if there's two thymidines next to each other, they actually will physically fuse together, and then your body gets confused. And then sometimes instead of just trying to stop fixing it, it will force its way through. And then sometimes when your body forces its way through, it can make mistakes. So that's how you can lead to genetic mutations from X-rays. This is the same idea from ultraviolet light, does the same exact chemistry, or very similar chemistry. Chemicals called mutagens, they can go in there and physically just mess up the DNA till your body tries and fixes it. Usually it fixes it, sometimes it doesn't, and that can cause mutations. And then some viruses can actually go in and change your genetic code. So if anyone has ever had like a cold sore or something like that from herpes, that's because herpes has gone and actually injected its own genome into your genome, right? So that's why you can get a cold sore, you know, 10, 20 years later after being infected, because the viral DNA is now part of your chromosomal DNA. So it can inject and change your DNA as well, which is another interesting side story too. They believe one of the reasons that allows for live birth in mammals is that there was a viral infection at some point along the evolutionary chain that caused basically the uterus to shut down the immune system, because the viruses will try to avoid the immune system. So there's no evidence of the virus, you know, creating viral particles, but usually during pregnancy, the uterus will shut down its own immune system because your body will actually try to kill the fetus because it thinks it's foreign. And there is the theory that's, I think it's currently well accepted. I know I was reading that in grad school, is that viral DNA from, you know, you're talking millions of years ago, actually can shut down the immune systems in localized spots. So cool little anecdotes. So types of mutations, right? The most common one is a substitution or a point mutation. So most, you know, things when they're bioengineered, scientists make very rational choices, and we usually do it by trying to change the least amount because it makes your life a lot easier. And so what a point mutation is, or a substitution, would be like that example I gave in the last slide when we had something like CAU was histidine, then we changed it to CAC and it became glutamine. The point mutation is changing that last part of the codon. So it's literally substituting a C in for a U. So we'll either call that a substitution or point change, right? A silent mutation would be something like, let me just double check so I don't give the wrong letter, would be, I believe this is true, it should be CAU and CAC, right? Yeah, I'm right on that one. Both, that is a point mutation, but both of these are codons for histidine. So it is a mutation because the U was mutated to a C, but when your body reads it out, it both turns into a histidine. So that would be a silent mutation. So technically mutated the DNA, but does not affect the function of the protein, right? And then a frame shift mutation would be if I had something like this, again, we'll take the CAU, right? And a frame shift would be the U still remains there. Instead of getting substituted in for a C, that would be like your body messing up and then jamming a C in there because it messed up replication. And so the sequence CAU is still there, but when you're reading through the codon now, right, you've changed it to CAC. I guess that's still histidine, so let's change that to something else. I don't know, I'll make it CA. What did I make it for? I don't know, make it up, right? If it's CAA, and you jam in the C, right, you're changing it from CAA to CAC. So it'd be a frame shift. So it's literally, right, you're reading every 3 nucleotides. Well, what you did is you shifted the frame that the amino acid was supposed to be in, because now you've shifted everything down by 1. So why don't we see a couple of pictures of what that looks like if we're looking at it like actual examples, right? So if we look here, this is going to be, they know there's a lot going on, so why don't we just take a look first before we make any mutations, right? So we have our DNA template on the top. mRNA is going to be transcribed from that template, and then what you can do is translate that into an amino acid sequence. So methionine, lysine, phenylalanine, glycine, and then you have your stop codon. Well, what happens, so I know it's kind of hard to see, so I'm going to go back and forth a couple times. So keep your eye out where it says substitution of C by T, right? So if you're taking a look where that is, right, there's our C right after the triple AAA, the one that says phenylalanine. That first C that I've highlighted now, right, we're saying we had a point mutation or a substitution of T into C. So what we've done now is we have a mutant protein. What did we do? Well, we changed the glycine, which is that last amino acid, into a serine now. And so that's a really big change. You've gone from a hydrophobic amino acid into a hydrophilic one. And that can actually substantially change the structure of a protein and occasionally kill it dead. So what about something like a frame shift mutation? So the example I gave you is that we inserted amino acid. It's just as common to lose lose a nucleotide. So before we had that AAA, which is encodes for phenylalanine. Well, it looks like that A got popped out, right? And I've made this mistake before making my own mutants, and it's not fun to find this mistake. So I've now mutated my phenylalanine into leucine. My glycine now becomes an alanine, and now my protein keeps going, right? There's no stop. And then I've accidentally done this before, screwed it up, and then your protein ends up like 50 amino acids long, and all that work you did for a week was useless, right? So that's on a bioengineered side of things, but your body does the same thing. And these can lead to bad mutations, obviously can lead to genetic diseases, cancer, occasionally can lead to good things, right? Very occasionally, but that's how evolution works. So how do we feel about types of mutations and what a mutation actually is, right? All right, so let's keep moving. So effects of mutations, I've been saying this a couple of times, right? You have your DNA, you have some sort of mutagen, it changes the DNA, that leads to a defective protein, and that defective protein can then lead to genetic diseases. And there's 2 types of genetic diseases, and it depends on where the mutations happen. So probably where most people are familiar with are mutations in somatic cells, which just basically means any cells that are just fundamentally yours, right? These will lead to cancer. Some genetic diseases can actually mutate in either the egg or the sperm cells, and if that happens, then they're actually hereditary, and they can be passed down to subsequent generations, and those will be called mutations inside germ cells. So there's only 2 germ cells, either sperm or egg. Everything else is going to be somatic because there's no way to pass on, like if you have, blood cancer or something like that from, maybe, or even put it this way, skin cancer, right? You tan too much, you end up with melanoma on your skin, you can't pass that genetically down to your descendants. If there was some kind of mutagen where it affected either the egg or the sperm cell, you can theoretically pass down that genetic mutation. Get in. There we go. All right, and now we're on our last slide, or last section. I did not, this is, if you check out the syllabus, it has always been, this was the last thing we were going to talk about. It just seems weirdly serendipitous, I don't know if that's the right word, that we're ending on this topic. So I'm keeping it pretty straightforward. I just really want to talk more, it's like, what are viruses? We're not really going to get deep into like COVID-19, especially because it's a new virus, right? But what are viruses, right? And so what is going to be fundamentally difference between like a human cell and a virus cell, or say a virus cell and a bacteria, right? Major differences is that we actually don't know, and you might have heard some weird conspiracy theories about this saying scientists don't even know what it is. No, we know what viruses are. We don't know if we can classify them as alive or not. Viruses are definitely biological. Are they living? That's open for debate, right? Bacteria 100% are living. Humans, cells, eukaryotes, living. Archaea, living. Viruses, we don't know, right? Typically, viruses are small, very small. I think COVID-19 is around 200 nanometers, so quite small, right? So they're either going to be between 3 to 200 genes. To put it into perspective, humans have anywhere between, I don't want to get this wrong, somewhere around 20,000 to 100,000 genes, right? Bacteria, usually much larger. I mean, some organisms are enormous, have a large amount of genes. Viruses are typically small. So we haven't really distinguished, okay, viruses have genetic information, they have genes, right? So doesn't that make them alive? Well, they can't live without a host cell. That's the first thing too, right? They need them to survive and to replicate, but then you might make the argument, well, don't some parasites require host cells? And that's true too. And they are alive, right? Major differences is that our viruses can either have DNA or RNA that's storing their information, right? Their genetic information. You cannot have both. It's either DNA or RNA. It actually helps distinguish what type of virus it is. And so COVID-19 has a RNA viral genome, right? It's usually surrounded in a protein coat. It's serendipitously lucky on this one. COVID-19 has one additional coating. It's actually surrounded in fat. That's why washing your hands works so well against it. And then that's why some disinfectants like isopropyl alcohol doesn't kill all viruses, but isopropyl alcohol is really good at ripping apart fat molecules, and that's why it can kill coronavirus, right? Major difference. This is the big one, right? Viruses do not have any of the necessary materials. That includes nucleotides or enzymes, or they don't even inside them don't have their own proteins, and they cannot grow. Everything is done inside the host cell. Essentially, the genetic information is injected into a host cell, and then it hijacks all of the material that's inside that cell. So it will hijack the ribosome. It will hijack other enzymes. It will hijack amino acids. It will hijack other enzymes. So it contains nothing to survive on its own. It absolutely must use the machinery inside a living cell. And the way that does this, that invades the host cell, that takes over the machinery, right? And then it steals any necessary proteins for synthesis. And then what it ends up doing is not really growth, it just self-assembles these particles. That's it. And then those particles eventually will just burst from the cell dies, and then the virus goes and infects a new host. So not quite alive, but not quite dead either, obviously, right? So what do you have up here is not a picture of a coronavirus. It's just what most people think, common viruses that you can see, right? So the idea from this picture here versus the coronavirus, as far as we know from electron microscopy, it's literally just a sphere with little spikes coming out of it, right? Literally, corona stands for crown. So it looks like a crown of spikes. So what ends up happening, right? And we can just use this other virus DNA here. This one's a DNA virus, right? It needs to collide with the cell wall. What it will do is going to inject its DNA or RNA into the host cell. From there, it takes over the host cell. It makes thousands to millions of copies of itself and eventually ruptures and then finds new uninfected cells and continues the infection that way. And so there's no drive. They're not evil or anything like that. They just do what they're programmed to do. That's it. There's nothing to them, right? Right, so actually everything I just wrote there is basically what I said, but let me just, read it off slowly to make sure everyone's on board. But right, we go in order of how the infection happens, right? So the virus enzyme makes a hole in the cell wall. And so you might have heard about the spike protein on coronavirus. this is responsible. And maybe if this is what you want to choose for your mini project, maybe, that one's definitely on the PDB. If you look at it, you'll actually see it looks like a needle. And that needle pierces into the outside of the cell, which we know is a fluid mosaic, so it can force its way in there. It will open up a hole, and then the DNA comes out through the virus, through that spike protein, and enters the cell. So then the viral nucleic acid will enter, it mixes with material in the host cell, and then the host begins to replicate viral DNA. So if the virus is an RNA, stores its genetic information as RNA, it actually has to do one step, which is what coronavirus would have to do first, is we'll actually do something called reverse transcriptase. So it will turn the RNA back into DNA, because that's how the human cells operate. So it's almost like going backwards a little. Or if it's just pure DNA, it goes in and just hijacks the cell. And then from there, it will produce more viral mRNA. And then it sometimes occasionally has only a handful of genes, right? So we'll have proteases, which are protein cleaving enzymes. They'll process the proteins. And then what they end up doing is just basically self-assembling. So there's not like, they're not thinking, it just happens through through interactions, right? But they'll self-assemble and eventually leave the cell by killing it, by erupting. And so I mentioned reverse transcriptase. I only wanted to mention this here. COVID-19 is not a retrovirus, because retroviruses technically they will include their genome into yours. So that's why like HIV is a retrovirus, herpes is a retrovirus. It will actually inject you and change your chromosomes. Coronavirus doesn't do that. But it will need to do something similar to what I'm describing here, where it needs to turn its viral RNA back into DNA, so basically the human cells can use it, right? And we'll use an enzyme called reverse transcriptase. So again, if we break down the name, right, transcriptase, ACE means it's an enzyme, transcript means it's doing transcription, right? So it's taking RNA to DNA or DNA to RNA, and it's saying it's going in the reverse direction. So canonically, we go DNA to RNA. So if we're doing reverse transcription, it's taking RNA, and then transcribing it back into DNA, right? And so once that's been done, then the host cell can take over, and then it will continue with its infection. So almost done. Or is that the last slide? I don't know. All right, this is the last slide. Then we're done. And we're 10 minutes early. Even better, right? It's a gorgeous day out. So actually, this is exactly everything that I just said, but in picture form, right? Virus goes in, reverse transcriptase makes viral DNA. DNA, if it is a retrovirus, COVID-19 is not, but something like HIV is, right? It will incorporate itself into the host DNA, and then from there, it can make more and more proteins. All right, guys, we're done. And we're done for good, I guess. The only thing we have coming up, right, is going to be our final, which is going to be on Saturday night. I know it's a weird time. I think it's 5 to 7. I'll have to double check that. My plan is, like I said, tonight I'm going to come up with the homework for you guys. I'll put up to try to either today or tomorrow get a review packet out so everyone's good on that. And I'll be holding a review session probably on, my guess is Friday afternoonish. I'm probably going to do both my 1102 and my 1109 review that day. And then depending on how people feel, maybe we might meet on Saturday morning. I just don't want to overwhelm people. Haley, I haven't decided on that yet. So she was asking, it's gonna be open notes in book 2. I need to discuss that a little bit more with some of the other professors, and I'll talk to the nursing professors, gotta make sure I'm on the same page with them. So I can't commit to that right now. I'm almost done putting together what it's gonna look like, but essentially it's just gonna be about 1.5 times longer than your normal exam. So you're talking about maybe 15 to 20 multiple choice, handful of short answers, maybe around, usually give you around 5, probably up to like 7-ish, maybe 7 or 8. And then instead of 1 long form question, it will be 2. And then you'll have the entire 2 hours to get it done. And the way I'm looking at it too right now, the way it's kind of shaping up, and again, I'm gonna put this all written down so you have it. Roughly, probably 2, basically it's the way that the course breaks down. I'd say about half of it will be Gen Chem, a quarter of it will be Orgo, and a quarter will be Biochem. Just the way the course just kind of generally breaks down. All right, any last questions, guys?
For the project, we were trying to upload it to EC Learn and there's no like submit button.
I thought I changed that on Friday. Maybe I hit the wrong thing. I'll do that right now.
Okay, thank you.
Yeah, I know it's a pain in the butt if you can't do that.
Wait, sorry, I know you definitely already said this, but it's due the 6th, right?
It is due the 6th, and I think I made it at 5 p.m. just so you just get it done type thing.
Okay, thank you.
Yeah, 6th at 5. So you guys still have 2 more days, 2 and change. All right. Anyone else? All right, everyone. We can leave. I know it's only about 5 minutes early, but it's a nice way to end the class. So I will follow up with all the stuff I said I'd follow up. Like I said, we'll have a review on Friday. Then we have our final on Saturday, and we're done. All right, guys. I will talk to you later. So anyone who wants to head out, you guys can head out now. I'll hang around.